This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 296, recorded on September 21st, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone. We have a beautiful day here in Charleston. It's a little fall-like. Woke up this morning. It was a beautiful 70 degrees with no humidity. So we've moved into Chamber of Commerce weather.
We have 22 Celsius and sunny here in New York City. It's pretty nice out, although I haven't looked outside lately. And as you know, there are no windows here at the incubator. If you enjoy our work in these podcasts, including This Week in Microbiology and many others, all under the Microbe TV banner, supported, of course, by the American Society for Microbiology, we'd love your support. And you can do that over at microbe.tv/contribute. Also, don't forget to check out Mark Martin's new podcast, Matters Microbial. I think he's up to episode number 7, and today releasing number 8. Always an interesting guest. Check it out, microbe.tv/mm. So I have a snippet for you today, which is based on a New York Times article by Carl Zimmer. And since many of you do not have subscriptions to the New York Times, in fact, I don't. I got mad at them last year and I canceled my subscription. Yeah, I didn't like their COVID coverage.
Okay, I can see that.
Which is, you shouldn't really do that because people can say what they want. And I get mad when people unsubscribe to us because they don't like what we say. So I'm going to resubscribe. Do you subscribe to the Times?
I subscribe to the New York Times and to my own hometown newspaper. So I'm an ardent supporter of the free press.
What about the Washington Post? Or
I subscribe to the New York Times, the Washington Post, and the Post and Courier here in Charleston.
Okay, no Wall Street Journal, right?
No Wall Street Journal. I used to get the Wall Street Journal, but it just became too much.
All right, I'm going to subscribe to the Times because I don't think it's right to get mad at a publication and then unsubscribe. I don't like when people do that to us, so I'm going to follow my own advice. And I thought a lot of people don't subscribe, so they'd like to hear a synopsis of this article. It was published on August 25th, about a month ago. It's called Researchers Dispute High-Profile Discoveries of Cancer Microbes. And the subhead is 3 studies in elite journals, found that tumors are littered with microbes, but several teams have been unable to replicate the work. All right, so the bottom line here is that over the past years, a number of people have found that tumors have a variety of bacteria, fungi, and viruses in them, and they seem to be unique to tumors and the tumor type, and they're not found in other tissues. And so that's a cool idea if it's true, because maybe you could diagnose cancers by looking at bacteria if they get in the blood, right? Or maybe you could treat tumors with antibiotics, and maybe they're dependent on the microbiome. So that's kind of an interesting angle to it. So the problem is there's a little bit of dispute, as sometimes happens in science, and that's what is covered in this article. So there's a paper published in 2019, the fungal microbiome, MYCO, fungi.
As for fungi.
Fungi.
Myco means fungi.
Promotes pancreatic oncogenesis via activation of MBL, which is mannose-binding lectin. This was published in Nature. And they basically find that they look at pancreatic cancers, pancreatic ductal adenocarcinoma, and they find a mycobiome distinct from that of the gut or normal pancreas. And this is all done by sequencing, right, ribosomal RNA, and using computational biology to order what's there and ask, is it really different from everywhere else?
And they're sampling the tumors. So what they are doing is they're sampling, if you will, a sanctuary where the microbe is either living or has actually become trapped
Right.
in that location. So it's not really, quote, normal host tissue. So one of the questions that you have to ask yourself is how good were they at sampling the area, and was there any contamination?
So this is several other papers have now said they don't believe this. They say the tumor microbes are most likely mirages or the result of contamination, as Michael put it more scientifically.
Well, you know, you don't know. I mean, often when you're going to resect a tumor, the first goal of the surgeon is to generate clean margins, and that is to get all of the tissue that's cancerous and go into healthy tissue so that you have, as they say, caught all of the adverse events that could then proceed and grow into new cancer.
Yeah.
And so the question is, what did they sample? And that you have to look at the papers, you have to look at the methods, you have to look at the quality control of the nucleic acid that they're recovering. And then most importantly, you look at the frequency of how much of the particular organism was recovered from the total nucleic acid fraction, that, and you compare it to an internal control. And so there's a lot of controls that are required to do this properly. And that was my only gripe with Mr. Zimmer's article, is he didn't really get into the inside baseball aspects of how hard it is. And I'm assuming that the peer-reviewed elite journals did a rigorous job of asking the questions I am raising.
Yeah, he just talks to people and they make comments like this one Steven Salzberg who has published a rebuttal from Johns Hopkins. He said they just found stuff that wasn't there because he didn't find it.
Yeah.
But other groups have found it. For example, there's a study out of Tokyo. They found fungi in pancreatic tumors from 78 out of 180 patients. There's a paper out of the Weizmann Institute published in Science. They looked at 1,500 tumors from 7 types of cancer, and they found that each one has a certain type of bacteria. But someone from Leiden University said they used the same methods they used in the Weizmann paper, and they could not find bacteria. He's quoted, "We do not find a single case where that's happening," and they published their findings as well. And then now the third study comes from someone we know from Rob Knight, who we have talked about and had here on TWiM. They published it. This is from UCSD, University of California, San Diego. They looked at a database of tumor DNA. It's called the Cancer Genome Atlas. And they asked the computational approaches to look at DNA sequences and ask, are there any bacterial sequences scattered among them? And they found them. The problem is they also found sequences from bacteria that infect seaweed, for example. So one person said, this is a sure sign that something is wrong, and deemed the seaweed bacteria and other out-of-place species nonsensical. And Dr. Salzberg has shown they've done the reanalysis of the same database, and they said it's wrong. And they published that in MBio. And then, so Rob Knight went back at his data and reanalyzed them. You know, part of the problem, as Michael said, could be contamination. The other problem is that if you leave human, if you don't get rid of all the human DNA, you could get complications as well. So,
and that's done by computational subtraction.
That's
so
right.
You may be throwing out the baby with the bathwater. because you may be throwing out some bacterial DNA in order to get rid of the human DNA. And that's why you have to look at the fraction of the microbial nucleic acid, whether it be fungi or bacterial. The other issue is we're often relying on 16S ribosomal RNA gene sequences to identify bacteria, not so much with viruses. And for the fungi, it's the intergenic region of their protein synthetic machinery, the ITS region, that is involved in identifying fungi in this targeted metagenomic sequencing, where they specifically just look for 16S ribosomal RNA and then extrapolate. So we don't know if the seaweed bacterium is related to a cancer bacterium, because the 16S RNA could be, in the same lineage, because not all bacteria, if one bacterium eats seaweed, another bacterium could be related to living in a cancerous cell based on the physiological conditions. And what's unique about seaweed is it's got that unique cell wall structure, which is why we use agar and Petri plates. So there's a lot of unanswered questions in these out of place, nonsensical species that are showing up. So
there may be a reason. There may be a reason for that, you're saying.
Yeah.
Right?
And we have to defend the pioneers who are out at the frontier trying to push the envelope, because what we know about pancreatic cancer is you got to catch it early, because oftentimes there are no symptoms until you're already in stage 4. And then that often tragically is a death sentence for the patient.
Well, Rob Knight and his group have defended their results. They say we still find this even though we've improved the analysis. And of course, people like to stand by their findings, right? They defend them. And the article ends with a quote from Arturo Casadeval, who has been on TWiM before. He's a fungus guy, right, Michael?
Absolutely. Cryptococcus.
He says, I would not be surprised if the disagreement causes both camps to innovate and push science further. This is a story of the scientific process at work. And I think that's a good quote, because that's the way it works, right?
And that is the principal conclusion from Mr. Zimmer's essay, is that this is a story of the scientific process at work. The first draft isn't always the correct draft. We iterate. Science is an iterative process. It requires probing, questioning, challenging, and we're witnessing the scientific process. And one thing I commend Mr. Zimmer for doing is he highlights the scientific process at work. I mean, he compares both sides. I mean, he has a few hyperbole comments in there about the seaweed and nonsensical, but at the same time, if you read it as I think it was intended by Carl, it really shows what Dr. Casadeval offered, this is a story of the scientific process at work.
Okay, so that's it for my snippet. Thought you would like to hear that story, which you probably can't read, and if you can read it, maybe that helps your comprehension of it. So, Michael, what do you
You can actually go to Carl Zimmer's website.
Yeah.
And this story is published. It's reprinted on his website in its entirety.
CarlZimmer.com. Wow,
Yeah.
Look at that.
And so you can read this essay.
Let's see. Here on the front page, writing, articles.
And it's in the articles tab.
Despite researchers dispute high-profile discoveries. There it is, right on his. I wonder how he does that.
He probably has an agreement with the New York Times that it's his work, so he can repost it.
Yeah, that's good agreement to have. Well, I'm going to put that in the show notes so that everyone can read it. Thank you, Michael.
Thank you, Vincent.
All right, what do you have for us, Michael?
I have something completely different. This paper comes to us from Nature. It was published on the 13th of September, and It's an open access paper, and it's by Nomura, Andreaza, Shang, Dong, Zhou, and He, and it's entitled Bacterial Pathogens Deliver Water and Solute Permeable Channels to Plant Cells. And the paper covers a topic I've always been fascinated by, namely the role that type 3 secretion plays in virulence, and in turn, the pathogenesis that a microbe can confer to the unsuspecting cell of its host. Now, as I said, this paper is open access. It's chock-a-block full of really neat experiments, and so I encourage you to go and look at it. But the first one, the reason it came to my attention is, to my knowledge, this is the first manuscript on TWiM where the first experiment of the results of the authors involves the formal use of artificial intelligence software. This is a AI product called AlphaFold 2, where the result from the in silico experiments then direct the authors into framing their actual wet work in order to assess the role that type 3 secretion plays in the pathogenic process. And this is a plant pathogen we're going to be talking about today. And the process is referred to as water soaking. That's a devastating, life-ending attribute in plants. So first up, for our new listeners, what is type 3 secretion? In its simplest form, the bacterium will deliver or inject a virulence determinant using a machine that looks remarkably similar to a flagellar motor assembly. This is the motor that spins the flagellum of a bacterium, and it's installed into the cytoplasmic membrane of the bacterium. And here, instead of spinning a flagellum at the end of the rotor apparatus, the bacterium exports proteins through a hollow protein tube that effectively expends proton motive force in order to push the protein from the cytoplasm of the bacterium into the cytosol of, in this case, it happens to be a plant cell. And they use this mechanism, and I'll put into the show notes actually what this little thing looks like. And, the flagellar rotor and the type 3 secretion system is really a remarkable microbial machine. The flagellar rotor, for those of you interested, may recall that it spins at about 6,000 RPMs or 100 hertz, which is the equivalent of 7.2 million proton fluxes per minute. So motility and protein export are energy-intensive processes. So this process of pushing proteins through this, if you will, nano needle into a host protein really should pay dividends for the bacterium. And the remarkable thing is, as you're pushing this effector protein from the cytoplasm of the microbe into the unsuspecting host cell, the effector protein that the bacterium makes inside its cytoplasm is never exposed to the external environment, where the effector molecule might be sensed and then potentially destroyed by the host defense system. Now, this is not a new observation, so you're maybe wondering, why is this in nature? There are many bacterial pathogens of animals and plants that employ a type 3 secretion system to get the host cell to do the bidding of the microbe. And since we're going to be talking about Pseudomonas and we're going to be talking about Erwinia, I'm going to drop into the show notes a review on type 3 secretion from Pseudomonas aeruginosa into our show notes. The trick with type 3 secretion lies in understanding how these effector proteins function in the host cell. For if we can figure that out, it will first help us to understand what's actually going on, and then help us to develop potential interventions that we might then be able to use to combat the mechanism as to how the pathogen is making the animal, or in this case, the plant cell, sick. Now, before this study, the research community largely pursued a working hypothesis that, like most other type 3 effector molecules in the family that we're looking at, are referred to as the AVRE DSPE family effectors, and they would target either host cell proteins, host cell RNA, or DNA processes in order to get the host cell to do something the host cell never intended to do, and they exert their virulence activities. Effectively, the bacterium sends in an instruction set that then results in the host becoming sick. So in today's paper, the authors are going to help us appreciate the precise mode of action of this widely conserved AVRE family effectors, including this role of DisBEE. And DisBEE is from the ubiquitous plant pathogen Racaniello amylivora, which is a native pathogen of wild rosaceous host. And if you're not familiar what a rosaceous host is, it happens to be an apple or a pear, crabapple, hawthorn, juneberry, serviceberry type of tree that is quite common in North America. And in fact, Racaniello amylivora was the first bacterium proven to be a pathogen of plants, and the common or the generic term for the type of illness this confers in pear trees is termed fire blight. And this infection affects the whole plant, principally, and this is the way I think about it, is it results in a form of diarrhea in the plant. Now, you heard that right. Plants get diarrhea. Now, the plant community refers to this as water soaking, so I'm sure I'm going to get letters. But here what happens is the blossom, well, it's true, Vincent. I mean, who ever heard of diarrhea in a plant?
Well, they're just losing their water, I guess, right?
And their nutrients. It's no different than us.
Yeah.
When those damn bacteria give us diarrhea, we're losing our electrolytes, we're losing osmotic tone. I mean, it's as devastating to us when we have diarrhea. And let's not forget, diarrhea kills a lot of infants globally. And water soaking kills a lot of plants. So this is not something to be trivialized. I'll also put in the show notes what fire blight looks like. But one of the most remarkable things about fire blight is you can actually see the diarrhea forming on the little tiny piece of fruit. And the ooze that comes out of the fruit is initially creamy, becoming amber tinged as the diarrhea effectively ages on the poor little fruit that's losing its water. The other plant pathogen that the authors look at is Pseudomonas syringae. This is another plant pathogen that invades damaged tissue and produces a toxin that kills surrounding cells where the bacteria can multiply. So again, we're going to have lysis of the apoplast, or, the plant's going to lose osmotic tone, it's going to lose solute, it's going to lose water. And Pseudomonas syringae, some of you may know, produces also a protein around which ice crystals can form. Mm-hmm. And as the ice crystals enlarge, they pierce and severely damage plant cells. And this is one of those cool experiments that often are done with both non-major and major students of both biology and microbiology. And again, into our show notes, I'm going to thank especially our TWiM adjacent occasional contributor, Dr. Mark Martin, for the very cool YouTube video on ice nucleation caused by this ice-producing protein from this particular microbe, Pseudomonas syringae. And it's a really cool video. You drop a few drops of Pseudomonas syringae into super chilled water, and it literally makes a brick of ice. So thanks to Mark for putting that video out on YouTube. So this shows what these proteins can do. So back to our story here. What do these two effector molecules, DisBe from Erwinia and AVRE from Pseudomonas, do? Well, the science community believed that they were involved in this induction behavior of water soaking. And again, this is what I'm calling diarrhea, which results in host cell death that are in turn conducive to bacterial multiplication with the infective tissues. And that's, after all, what salmonella does in our gut. The salmonella multiply when they give us the diarrhea. And that's what happens here with Erwinia in water soaking, as well as Pseudomonas syringae. But the rub is how these effector molecules are able to carry out this phenomenon, or more directly, how are they going to accomplish this feat biochemically? Now, the authors offer that the precise biochemical function of the AVRE family of effectors has been recalcitrant to sufficiently granular or mechanistic understanding for well over 3 decades. And one of the likely reasons has been attributed to their extensively large size. These are big proteins. They're about 2,000 amino acids in size. And a protein of that size, there are many domains that can interact with the host, but here none have any particular clear function. Second, there's the fact that these proteins are toxic when overexpressed in yeast or plant cells, and they are not all that similar to proteins in the database. And the literature has offered very few hints in that when they are expressed, the effectors moved or were trafficked to the cell membrane after their injection in the plant cell. And the AVRE DisBe family of effectors, we know, are essential for many plant pathogens to cause this water soaking disease. That is to say, if you knock out the expression of either AVRE or DisBe in the respective bacteria, the bacterium is no longer pathogenic to the plant. Now, this family of effectors is also uniquely associated with macroscopic water soaking of infected plant tissue. And as I often said, you don't need an MD to diagnose diarrhea. Everybody knows it when they see it. And water soaking, any plant anatomist immediately recognizes it when they see it. And water soaking has an active role in modulating the hydration of what is termed the apoplast, which is nothing more than an operational definition of the space outside of the plasma membrane of the plant cell that allows then free movement of material, solvents across the plant tissue. Today, in their papers, the authors first use this AI-based AlphaFold 2 software to effectively in silically fold AVRE family of effector proteins. And what they determined is that AVRE, this AVRE family, folds into a beta barrel structure that resembles a bacterial porin. So what's a porin? Well, porins are typically a bacterial structure, generally in gram-negative microbes, that are involved in the exchange of nutrients. They let things in and they push things out. And they're generally in the outer membrane of gram-negative bacteria. And porins are also been implicated in the pathogenesis in many gram-negatives. And if you think about it, that's what water soaking is things are moving in and out. So the authors used this artificial intelligence to test whether the unexpected prediction was correct by performing targeted studies. Now, this is not in silico. This is actually truly wet work to assess whether the proteins had indeed or do indeed form a porin-like structure or channel. And they principally asked the question, are they making a channel? And the experiments that the authors did, they demonstrated that the AVRE family of effectors did in fact fold into a beta barrel structure by using this ingenious model system. They use Xenopus oocytes, which facilitates the study of complex biological processes within a cellular context through the expression of exogenous mRNA and the concomitant expression of the protein. And what they learned is that the expression of AVRE and DisBE and these Xenopus oocytes resulted in both inward and outward current flow. So they're measuring voltage, if you will, using patch clamp technology. And again, this is an open access, so you can get deep into the weeds to see how they did this. This is a great paper in answering the fundamental questions of science. How do you know, and how did they show? And they do it beautifully. And what they saw, in addition to the inward and outward current flow, they saw permeability to water and osmolarity dependent oocyte swelling and bursting. Then, in another clever experiment that involved liposomes, which is nothing more than a glorified set of membranes, they reconstructed the DisBE channel or porin, and they asked the question, was the porin sufficient to allow the passage of small molecules? And the small molecule they used was fluorescein dye. And as you know, fluorescein dyes fluoresce. So by demonstrating that the effector expression in the oocyte promoted the flow of ions across the cell membrane, as well as swelling and burstings of the cells dependent upon solute concentration, which they measure as osmolarity, and passive uptake of fluorescent dyes, all of which are consistent with the behavior of a channel or a porin. Then in the ultimate how do you know, how do you show experiment, they offered that the presence of the effector protein in the liposomes containing the fluorescent dye resulted in the release of the dye, and the liposomes containing a fluorescent protein that was larger than the predicted size of the channel, which is 15 to 20 angstroms, or those of you who don't remember the conversion to angstroms to nanometers, 15 to 20 angstroms is 1.5 to 2 nanometers. It effectively blocked it. It didn't get out. And now the critical step in all of anytime you do experiments where you're trying to answer about pathogenesis, they ask the age-old question, what can you do to limit the severity of the infection? And what they did is they used a chemical blocker that would effectively plug the leak. The compound they identified was polyamidoamine, and it was a dendrimer that for all intents and purposes looks remarkably like a snowflake. And once you see the structure, and again, I'll drop a picture of what these dendrimers look like into the show notes, you will appreciate how it might function to simply plug the pore. And that's precisely what it does, as the polyamidoamines were found to broadly inhibit the AVRE and DisBE virulence activities in first the Xenopus oocyte model system, and then during a real infection caused by both Erwinia as well as Pseudomonas. And they have model systems for Erwinia. They use little pear fruitlets, and there's a picture of these little pear fruitlets in the manuscript. And it's a fascinating fascinating experiment where they took two bacterial pathogens and then via type 3 delivery of either the AVRE from Pseudomonas syringae, the cultivar tomato, on the model thalcrest plant, which is Arabidopsis thalinea, and the Dispe channel former from Owinia mellivora, which is fire blight on pears. They literally stopped the water soaking activity by simply administering these dendrimers, these amino dendrimers. And they illustrate this all quite nicely in, of all places, their extended data figure 8. And it's a clear model system for the molecular actions of what AVRE is actually doing in this plant. Now, the wonder of all of this is that the bacterium is injecting a protein into the plant cell that is effectively poking a hole inside the plant cell to allow its innards to wash out to effectively feed the bacterium without having to, get rid of the cellulosic cell wall of the plant cell. It's literally poking a hole in it.
So, is that what it's for, Michael? So the bacterium can grow?
Yes. Yeah. It answers the age-old question, why do bacteria infect us? And the answer is they're out to get food. So what they have done in this manuscript is they've unraveled a new type of type 3 secretion attribute. Namely, the bacterium injects a protein that assembles into a beta barrel porin-like structure that literally confers an equivalent of in situ diarrhea that our plant physiologist refers to as water soaking within the plant, or the apoplast, that in turn results in solving the conundrum, why do bacteria infect hosts? The answer being they're simply out to get food. They close by offering that future research is needed to understand how this AVRE family channel activity mediates water and solute and nutrient flows with respect to the apoplast's osmolarity, and to generate this macroscopic water soaking behavior. We're literally seeing, water accumulated on the surface of the piece of fruit. The authors close by offering that as large proteins with potentially many protein interacting interfaces, potentially with the host, the AVRE family of effectors can likely additionally engage these other host proteins, including plant protein phosphatases, PP2A, one type of protein phosphatase, and a receptor-like kinase to affect other aspects of AVRE Dispe function. So as our former colleague Ilya Schechter would often say, stay tuned, there's more to come, probably from this family of effector molecules that are responsible for this water soaking behavior.
So these compounds that plug it, they could be therapeutically useful.
You could imagine spraying it on your fruit trees if it's infected with Erwinia, which is a pretty devastating plant pathogen because it can take out not only your fruit crop, but it can actually kill the tree. And so, these are nothing more than grease that they're spraying on it, and it's plugging the hole. So the discovery of these water insoluble permeable channel functions of AVRE family effectors has broad implications for the study of bacterial pathogenesis and bacterial disease control in plants. And I was talking to one of my colleagues, Joe John, about this paper, and he was saying what this reminds him of is homolysins that are produced by bacteria that literally cause the lysis of red blood cells. And any first-year micro student has seen the effect of homolysin on a sheep red blood cell. It effectively clears it. It lyses it. And that's what we're witnessing here in the plant cell. So it's really a quite remarkable behavior that we're witnessing. And as I said, the authors really take us to school teaching us how to to measure these activities. Their data are quite compelling, and they're doing it with voltage clamping. And it's really a very cool paper for you that I'd like to call to your attention.
So these compounds that the PAMAMs that plug the channel, they just pick those based on the diameter of the channel.
Yes.
Pick something that they thought would block it, and it turned out to do it, right?
Yeah, it's fascinating.
It's amazing.
Well, I think they knew a little chemistry too. This was not for the uninitiated.
Yeah. The beauty of this paper is it takes you from, you have these genes, and you don't know what the product is. You use the AlphaFold to give you a prediction. You say, oh my gosh, it's a pore. And then you actually do the experiment in Xenopus to show it's a pore. And then finally you do it in plants and you show it gives them diarrhea. And
modern Koch's postulates.
And then you can inhibit it on top of that. It's just the perfect story, right? It's beautiful.
Yeah, I really enjoyed this paper.
Very nice. And it's a example of how everything lines up and goes really well. And so my hats off to the authors for doing this. And there are only 1, 2, 3, 4, 5, 6 authors. It's really good.
Yes.
All right. Thank you, Michael. Let me read a couple of emails here from Martha. Subduction. The topic has been on my mind since my recent trip to the Philippines, located in the Ring of Fire. My little guest hut was in sight of an active volcano, and the surrounding topography is unlike any I have seen. I have been wondering what part subduction played in the formation of the landscape. So the first paper discussed regarding microbiogeography to tectonics and teeth introduced an additional fascinating layer of questions to my existing questions about subduction. I never considered what the initiation event of plate tectonics might be. Regretfully, I had never studied enough chemistry to appreciate that such an event could be biological in nature, with the iron spewed from a volcano being the energy source for microbes. Wow, thanks for bringing this to my attention. There were so many new ideas that I listened 4 times, took notes, and now have a list of questions for my geologist friend. Then there was the paper about teeth. Fascinating. Dr. Schmidt, you kindly invited me to suggest topics of areas of interest for future discussions. I know this is very broad, but as a low country resident and avid kayaker, if you ever come across a paper relating to water or other wonders of our beautiful coastal environment, I would love to learn more. Who knows what you might turn up. Last week, I met a cell biologist researching elasmobranchs in my coffee shop. He answered my question about a small shark that I had observed a few days earlier. Serendipity. Many thanks to you and Vincent for making science education more accessible to the rest of us. Well, today we had a water paper, Michael. It's perfect, right?
Yeah, well, yeah, absolutely.
Alfonso writes, Dear TWiM team, my child is preparing a biology work in grade 10 about terraforming Mars.
Oh, my.
They have not been able to find sources for Azotobacter binary fission rates in vitro in order to estimate the potential reproduction rate in the ship journey from Earth to Mars. Would you be able to provide a source or a direction on how to find it? We already made multiple searches in PubMed, NCBI, Microbiology Society, ASM, scientific journals, and we are stuck. Alfonso, I cannot help you. What about you, Michael?
I looked at this, and I too did the quick and dirty search through the usual suspects. And I think what they're asking for is the generation time of Azotobacter. And it depends, because Azotobacter is one of these organisms that's out there, and it makes a tremendous amount of extracellular matrix. And it's actually used for drilling mud, of all things, because it's so slippery. And I don't ever recall anyone talking about its generation time. So I'll have to put that on my list of things to investigate. So thank you for the question.
I am right now sending your text as a text to Mark Martin. And maybe Mark will be able to know. So there you go. I sent it to Mark. We'll see what he does. And finally, Lisa writes, June 9th, TWiM, familial adenomatous polyposis runs in my family, and it has been highly variable how much the gene has caused disease in different family members. And my impression from when I have read about the disease is that our experience is in line with the general pattern. Some people with the gene for FAP have many polyps and many of the other possible symptoms, and some have almost none, even if they live to be 90. And thinking about the research you discussed explains part of the variability. TWiM, what was that episode? Do you remember, Mark?
I don't recall.
Let's see.
It was just before ASM. It was the one immediately before Mimi.
Oh, here it is. TWiM 171. Colonic biofilms containing tumorigenic bacteria in patients with colorectal polyps. There you go.
And that ties in with today's paper, that the essay by Carl Zimmer.
It sure does. Patients with familial adenomatous polyposis harbor colonic biofilms containing tumorigenic bacteria. There you go.
Well, that is indeed. We know that there are cancer-causing bacteria in our gut. They effectively transform bile acids into carcinogens. Right. And that in turn, if you happen to have a P53 lesion in your genome, makes you more susceptible to developing adenocarcinoma.
Right.
And all of that is being investigated by many, many cancer labs.
All right, that'll do it for TWiM 296. Show notes, microbe.tv/TWiM. As I said, we'd love your financial support, microbe.tv/contribute, and send your questions and comments to TWiM at microbe.tv. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent. Enjoy your week.
Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.